Good morning. Delighted to be here to share with you the tremendous progress that Orchard Therapeutics has made just 2 years since its inception in building out a portfolio of gene therapy, ex-vivo lentiviral gene therapy programs for a variety of rare diseases. So our mission is to transform the lives of patients with rare diseases through innovative gene therapy. And I find the use of that word transform something that we don't want to undervalue. It's an extraordinary thing, I think, to be able to use that word today. I've spent 19 years, the last 19 years, launching orphan drugs for rare diseases. And in all these cases, it's been about slowing disease progression and trying to improve quality of life. But we're at this new era where there's a real possibility of giving patients normal lives, and that really is amazing. And our vision as a company is to be a fully integrated global biotech in this gene therapy arena that brings these patients these transformative medicines. And again, my experience, 28 years in the industry and 19 years in rare diseases, is that typically it's smaller companies of our size that can really deliver to these small patient populations around the world, where you need tremendous interconnectivity amongst the, with patient organizations, with thought leaders, and of course with the science. So we're gearing up to be one of those companies. And I'm privileged, having taken over the helm a couple of months ago, to have inherited a leadership team that collectively has over 80 years of experience in gene therapy. This is a new arena, but this team has spent a lot of time in this arena. So we were incorporated in September a couple of years ago, and a great deal has happened in this time. So you can see that really back in 2016, from a program and portfolio perspective, we were founded with licenses with UCLA, University of California, Los Angeles, Boston Children's Hospital, and others, with our first indication of ADA-SCID. Followed quickly with a license with Manchester for MPS-IIIA. So these are very rare genetic diseases that maybe some of you know. We've also added to the pipeline with additional licenses very recently, and I'm not yet in the liberty to share that with you, but at one stage in the near future, I hope to show you our whole portfolio. We've been raising money in this period. We had a Series A of 30 million. We had a grant of 90 million from the Californian Institute of Regenerative Medicine. And then we're in late stages in a Series B financing, which we're looking forward to closing in the not too distant future and sharing that news with you. Also importantly, we've been building our commercial manufacturing platform. As you'll see, one of our challenges has been that the clinical data is well ahead of our commercialization capability. So we've been trying to catch up to be able to bring these ex-vivo lentiviral gene therapies to patients as quickly as we can. So I'll spend a little bit of time in a minute talking about the ADA-SCID program, and I'll say a few words about the MPS-IIIA program. But, that portfolio has grown considerably, and we'll talk about the 5 to 10 programs that we now have in the near future. And one of the key drivers of this pipeline has been the exquisite nature of the collaboration between our company and academia. And I think that so much of this foundational work has been done in leading global centers, as you see here, whether it's in the field of primary immune deficiencies, with places like UCL, Great Ormond Street, UCLA, Boston Children's Hospital, and the NIH. These are members of our scientific advisory board, or in metabolic disorders, Manchester, and again, Boston Children's Hospital. So with this group, what we're able to do is really de-risk programs by looking at the preclinical proof of concept with academia, as well as even going into clinical proof of concept before you go all the way to commercial stage development and registration studies. So we think it's a very good symbiotic relationship. So I'd give you the example of ADA-SCID. So as you know, this is a very devastating primary immune deficiency where you lack an immune response, and untreated kids will die, under the age of 1 typically. Now there are alternatives available today, but we believe that ex-vivo lentiviral gene therapy offers a great opportunity to try and normalize the lives of these patients. So this was some data that goes back to March of 2016, when there were about 33 patients already studied for up to around 4 years in 2 sites at the University of California, Los Angeles, and UCL Great Ormond Street. And what you can see, which is really remarkable, is 100% survival rate, and collectively over 93% event-free survival. And that's in that initial cohort. So this is not the kind of data that I'm used to, having launched enzyme replacement therapies, other types of rare disease product. This is unusual. And you see that response based on also a recovery of the immune system, whether you're looking at T cell recovery, B cell recovery, or NK cell recovery. And that recovery is the same whether you're at UCLA or here in the UK. So going forwards now to June of this year, where we took a further cut of the data, and we're now at nearly 50 patients, we continue to see 100% survival for over 5 years, and we see 98% event-free survival. So again, these are very compelling numbers, and we feel now a great sense of responsibility to bring this therapy to patients around the world as quickly as we can. And with that in mind, we'll be filing our BLA with the FDA by the end of next year. Now turning to MPS-IIIA, some of you will know that this is caused by an absence of an enzyme which leads to substrate development in the brain and other parts of the body that causes severe neurological and motor impairment. And untreated, these patients will die in their teens and 20s, and there really isn't treatment available for them today. So we have mouse model data that's been generated through our collaboration with academia. If you look at the MPS-IIIA mouse, on the left-hand side, you only see 3% of this essential enzyme being generated in the brain. With CD11b vector gene therapy, you see about 11%. Now, that's not the same as wild type, but actually it's sufficient to, in the mouse model, deliver benefit and reduction of substrate. So you can see in the next part of the slide about an 80% reduction in substrate in the lentiviral gene therapy treated mouse relative to MPS-IIIA, and you also see a response, if you like, in the clinical sense with the mouse, where you see reduced hyperreactivity. What is encouraging is that similar data was produced for metachromatic leukodystrophy, where this kind of data led to good clinical outcomes in patients. So over 20 patients have been treated with MLD on the basis of similar mouse data. And so we feel confident about going into the clinic with this kind of response. So I want to just finish by saying, one of the challenges I say is that we have a growing body of clinical evidence in a number of programs, and now we're trying to fill in by developing our commercialization capability, putting in our supply chain and manufacturing platform, with Oxford Biomedica for vector supply, with Lonza PharmaCell and PCT doing the cell manufacturing, so that both in the United States and in Europe and eventually other parts of the world, we can bring this treatment to patients and help transform lives as gene therapy has the potential to do. Thank you very much. If I have time for a question, I will take one. Oh, versus Strimvelis? Well, firstly, I would say that Strimvelis represents a tremendous breakthrough, we're, very honored, if you like, to be following in those footsteps, because it really was setting a new trajectory for this type of medicine. I think one thing that you will see here is in that data a tremendous response, both in terms of 100% survival rate, but also the event-free survival rate is very high. And I think that is something that I'm sure patients and families will look at very carefully. I think the other thing that you will notice is that we're going down the cryopreservation route, so that it means that patients will be able to be infused at their home infusion center and won't need to travel in order to get treated to Italy. I think it's one of the differentiating factors, but I think it's an important one from a practical point of view, because to ask families and patients to go to other countries to get treated commercially is a tough proposition. And I think ideally you want to be able to treat them as close to home as you can. And then I think from a reimbursement point of view, it's also trickier to do it that way. I'm not saying it's impossible, but I think it is easier to manage the reimbursement in your home country. Thank you very much.